This is the Kreb Cycle by
Adrian,
Emily,
and Dylan. Step 0. We just got pyruvate from glycolysis, and pyruvate dehydrogenase is going to catalyze a 3-step mechanism for decarboxylation, then oxidation, and then transfer of coenzyme A to make acetyl coenzyme A and a byproduct of CO2 with the help of an electron carrier molecule NAD plus to NADH. Step 1. The formation of citrate. With oxaloacetate, acetyl coenzyme A from step 0, and water, citrate synthase, the enzyme, is going to convert these into citrate and a coenzyme A, the thiol group. Step 2. The formation of isocitrate. Citrate from step 1 is going to act with aconitase, the enzyme for step 2, and aconitase will first do a dehydration event to form cis-aconitate. It will then do a rehydration or a hydration event to form D-isocitrate.
Step 3 of the Krebs cycle is the conversion of isocitrate to alpha-ketoglutarate via the enzyme isocitrate dehydrogenase. This is going to proceed in 2 steps, the first one being the oxidation of isocitrate, which is going to produce our first NADH molecule of the Krebs cycle. The second step is the decarboxylation, which will produce one CO2 molecule. The Krebs cycle is the conversion of alpha-ketoglutarate to succinyl-CoA by the alpha-ketoglutarate dehydrogenase. This reaction is in 3 steps. First, the decarboxylation of alpha-ketoglutarate, then the reduction of one more NAD to NADH, and the subsequent transfer of CoA, which forms the end product succinyl-CoA. The alpha-ketoglutarate dehydrogenase is a key control point in the Krebs cycle. It is inhibited by the products succinyl-CoA and NADH. A high energy charge in the cell will also be inhibited. ADP and calcium ions are allosteric activators of the enzyme. Step 5 of the Krebs cycle is the conversion of succinyl-CoA to succinate. This is done by succinyl coenzyme A synthetase. This step is a 3-step mechanism. The first step involves displacement of CoA from succinyl-CoA by a nucleophilic inorganic phosphate molecule to form succinyl phosphate. The enzyme utilizes a histidine residue to remove the phosphate group from succinyl phosphate and generates succinate. Finally, the phosphorylated histidine transfers the phosphate group to a nucleoside diphosphate, which is our GDP, forming our GTP.
Step 6. Formation of fumarate. This step is a redox reaction that oxidizes succinate into fumarate while abstracting 2 protons and a pair of electrons, reducing FAD to FADH2. Succinate dehydrogenase, the enzyme that catalyzes this step, is bound to the inner mitochondrial membrane. It's an iron-sulfur protein that uses the covalently bound FAD to extract 2 protons to form FADH2. The FADH2 remains bound to the enzyme that passes the electrons onto the iron-sulfur component of the enzyme, which passes it across the electron transport chain. Step 7. Formation of malate. Fumarase catalyzes the hydration of fumarate to malate. In this step, water attacks a specific site so that the L isomer of malate is formed. Step 8. Formation of oxaloacetate. Using malate dehydrogenase, we oxidize malate into oxaloacetate. Using the electron carrier NAD+, we get NADH plus a proton.
In conclusion, because the Krebs cycle occurs 2 times for every glucose molecule that enters glycolysis, the input is 2 pyruvate, 2 ADP plus 2 phosphates, 6 NAD pluses, 2 FADs, and the total output is 4 carbon dioxides, 2 ATP, 6 NADH, and 2 FADH2 molecules.